Good afternoon. So I was asked to contribute with a lecture on a basic science technique which is related to the research topic that we have in Project 4. So I'm going to be talking about gene expression profiling, and the presentation will be basically divided in two parts. A first part that will present the techniques, the basic techniques of gene expression profiling. I will cover the traditional techniques such as DNA microarray and real-time PCR, but I want also to touch on some newer techniques like RNA-Seq that are revolutionizing the field. And the second part of the talk will be focused on the application of some of these techniques to xenotransplantation. I will not be covering specifically gene targeting. Gene targeting can be done, of course, in our field by genetically modifying the pigs, and the next speaker, Simon Robson, will talk a little bit about transgenesis in the pig. And as you probably know, another way to target genes would be to use siRNAs to modulate gene expression in an animal, and that is becoming now possible, although still difficult. But we'll talk about the analytic techniques for today. So gene expression profiling is the approach which consists in looking at tens or hundreds or thousands of genes that characterize the activity of a cell, whether a cell is getting activated, treated, whatever stimulus is applied to a cell, it changes its activity, its transcriptional activity. And this results in a change in its mRNA content, in its transcripts content. And so gene expression profiling is the activity which measures those changes. And it's typically defined as transcriptomics. Now it has been defined as transcriptomics. It is, in the big picture, if you want, a part of genomics approaches, although genomics per se is in the analysis of the genome at the DNA level. And it's complementary to other approaches looking at cellular activity, either at the protein levels by proteomics approaches, at the cellular level by other cellular assays. And there are also other omics now that people are looking at, either as biomarkers or mechanistically, such as metabolomics, lipidomics. So there are several ways that one can interrogate a cellular activity. So again, we're going to look specifically at this part here, the transcriptomics part. So basically, we want to know whether there is a change in the abundance of mRNA, so transcript levels in a cell in a certain condition. So basically, what is the transcriptional activity going on in that cell? There is a direct relationship, of course, as you know, between transcriptional activity and protein activity, since transcripts are translated in proteins. But this is also a process that's highly regulated. So the traditional approach, high throughput approach that has been used to profile gene expression is DNA microarray analysis. And there are newer techniques, and we'll talk about the RNA-Seq technique next. The microarray approach consists in transforming mRNA molecules in complementary cDNA molecules by reverse transcription, labeling them with a dye and applying this population of labeled DNA to a chip that is coated with a small oligonucleotide called probes, and then looking at intensity of signal on each of these probes by fluorescence. Now it's detailed here. So here we start with a sample from which mRNA is isolated. mRNA is reverse transcribed in cDNA, labeled to either Cy5 or Cy3, hybridized to the chip, washed, then the chip is scanned, and the fluorescence is measured analytically. There are different types of chips, biochips available. The traditional chips are manufactured by Affymetrix, and they typically contain probes that cover the entire genome of the species of interest, whether human genome, mouse genome, and they now have a pig chip. But there are other types of arrays, and I will show you an example of a smaller array that was custom designed in Simon Rapson's study. The experimental design is really important and really key in this type of experiments. It's a very powerful technique, but how you think about the technique before doing anything is key and really, really important. Typically, not always, but typically, one thinks about a study sample and a normal or control sample, whether it's a normal and a cancer cell or resting and an activated cell, etc. RNA is isolated from both samples, cDNA is generated, and then each sample is labeled with a different color, with a different fluorochrome. Then those populations are combined and hybridized to the microarray so that one can look at presence of one sample relative to the other sample. So this is a typical two-color analysis. Another way would be to only look at one color at a time and then across experiments compare expression levels between samples. But this requires more standardization, and it's more difficult to look at absolute quantities, and more controls basically are needed because there is no internal control. Biological replicates are very important, and technical replicates are important. They are both needed, and maybe we can spend a little time on the difference between the two. A technical replicate is to basically take mRNA from the same sample and hybridize it to two different chips. And so comparing two different runs from the same sample will give an idea of the variability due to the technique. Biological replicates will indicate whether there is variability between cells or between runs. So cancer cell could be dependent on the culture conditions, for example, and so testing the difference between two cell types on two different days in two different independent cultures will confirm any difference that could be seen on the first run, and so this would be a biological replicate. And the number of genes that can be studied can be large, relatively very large, with the whole genome ships from Affymetrix, or rather limited if one only wants to interrogate a few genes. The other very, very important feature in gene expression profiling is data management. It's absolutely key to have a good approach and good tools to analyze the results that are generated. Basically, this is a technique that generates lots and lots of data. And there are several steps in the analysis of these results. Until one can see the results, one has to go through several tests, for several steps, very rigorously to be able to obtain valid data at the end. There is a normalization step to subtract background from each sample. There is a filtering step to basically define a threshold and a level of positivity. And then there are basically two approaches to the analysis. There is a so-called unsupervised analysis and a supervised analysis. The unsupervised analysis doesn't provide any hypothesis as to what differences are expected between the samples and the controls. And so it's a statistical comparison between the groups. Whereas the supervised analysis is using some type of filtering to look at the results, at the data. So this DNA microarray technique has been used for the last decade to profile gene expression in various samples, both clinical and experimental samples. Very recently, in the last few years, a new technique has been developed, which is called RNA-Seq. I find the name a little bit misleading. It is suggesting that RNA is sequenced, but in fact, mRNA is still reverse transcribed in cDNA. And then this cDNA is fragmented. Libraries are made either of large or small molecules. And then those molecules are sequenced. It's a high throughput parallel sequences of everything that is contained in the sample. The sequences are then aligned to a reference genome. So if one is working with human samples, the sequences are aligned to the human genome. If it's a mouse sequence, a mouse sample, mouse genome, and it has not been done for a pig sample as far as I know. So I incorporated a few slides to give you an idea of what is possible with this RNA-Seq approach. Here, this is a paper that I found really interesting, in which they looked at the role of how RNA-Seq can be used to look at gene fusion in cancer. So they here show the various mechanisms by which genes can fuse. And in this very nice paper published in Nature in 2009, they describe how RNA-Seq can identify gene fusion in cancer. This slide shows a typical RNA-Seq experiment, which starts with the isolation of RNA. Some people choose to work with mRNA by isolating messenger RNA using a poly-DT tail that will anneal to the poly-A tail and purify mRNA. But many techniques just use total RNA after depletion of the ribosomal RNA. So if you isolate total RNA from a cell, about 80% majority of the RNA molecules will be ribosomal RNA, and they will basically create background in this assay. So ribosomal RNA is depleted, and then what's left can be studied with this method. Our mRNA is transcribed in cDNA, libraries are constructed, and then the sequences are read. They are read multiple times so that the sequences can be correlated and cross-check. These are called reads. And after maybe 5 to 10 reads for each sequence, the sequence can be derived and then aligned to the genome. So the chemistry and the mathematical approach behind this is fairly new and still evolving and complex, so it's beyond the scope of this lecture to go through all these details. But I wanted you to be aware of this technique, and I can provide you with references if you're interested to read about it or apply it to your system. Here are some points of comparison between RNA-Seq and DNA microarray. They are basically both high throughput sequencing to some extent. The resolution is much higher with RNA-Seq. It's at the level of a single base versus several to 100 base pairs for an array analysis. So throughput is high. We're comparing the first and the third column here. With the DNA array, we are relying on the genomic sequence, of course, which is providing the sequence for the probes that are actually put on the glass, versus here, we don't rely on any sequence. Basically, everything which is in the tube is sequenced. And then one can try to correlate it with something. There is less noise, a very good dynamic range. And what is really the plus of this method is that it can identify different isoforms, allelic expression. It's very sensitive. And what is not maybe emphasized here is that it's quite quantitative. It's more quantitative than an array analysis. And as far as the cost, I'm not sure it is really true that it's that low, but that was for RNA-Seq. Now, real-time PCR is a completely different approach. So it's not a high throughput approach, and it's not a screening approach. It's basically the opposite, where you decide to look at a few genes, and though you can look at up to hundreds of genes, but not thousands, definitely, with PCR. In a very focused fashion, and because of this focused approach and the nature of PCR, this is a very quantitative approach. It's complementary to the array and the RNA-Seq approach, if you want, in that in a workflow you would have a first screening with a high throughput approach, either array or RNA-Seq, and then validate the most interesting findings with real-time PCR and maybe quantify them more precisely. So it's basically a PCR-based approach, which you're probably familiar with. Is a chemical reaction that duplicates DNA by using the DNA polymerase, Taq polymerase. It's very versatile. It can be used on different samples, whether it's tissue, cells, fresh, frozen. It can be used on total RNA or mRNA. It can be used to look at one target at a time, multiple targets. And if the quantity of RNA is very limited, the RNA can be amplified prior to the PCR, either in a gene-specific fashion or in a non-specific fashion. The reverse transcriptase can be done using specific primers or random primers or oligo-DT primers. By the way, if you have questions, don't hesitate to stop me, because I'm not sure if I'm giving too many details or not enough at this point. So after reverse transcription, there are different enzymes that can be chosen from to reverse transcribe RNA. The cDNA is tested, and basically the real-time PCR means that the PCR reaction is monitored in real time as it goes, as opposed to the traditional PCR, which is looking at the result, the produced DNA at the end of the reaction as an endpoint reaction. So it gives you a much more quantitative analysis to look at the reaction real time as it goes. Usually, it's done in one step, in two steps, where RNA is reverse transcribed in cDNA, and then cDNA is amplified during the PCR. But some protocols and some reagents are developed in which this can be done in one step. So there are basically two ways to detect DNA as it is produced during the PCR reaction. The first and simplest way, and this is one of the earliest way that we've been using for real-time PCR, is to add a dye to the reaction, like cybergreen, which will intercalate into DNA bases. And as the DNA is duplicated, the number of molecules, of course, will increase, and there will be more dye bound to DNA, and therefore more fluorescence. So the signal will increase as DNA is produced. This is a very cheap and easy way to detect DNA as it is produced. However, it has some drawbacks. One is that the reaction is only dependent, the reaction and the detection is only dependent on the specificity of the primers. So we use two primers, a reverse and a forward primers, binding to the 3 and the 5 prime ends of the DNA strands. And each primer will anneal with the complementary strand of DNA. Taq polymerase comes in and it elongates DNA, and the specificity of the reaction is only strictly dependent on this small portion of these maybe 10 or 20 nucleotides that anneal with the target DNA. So specificity has to be checked. And then the other problem, which sometimes cannot really be solved for some of the targets, is that primers can anneal to each other instead of annealing to the target DNA. And because they then form double-stranded DNA, the dye can bind to the primer dimers themselves. And that creates background in the reaction that is detected as signal. But of course, that's a wrong signal. It's a non-specific signal. So I showed you one of the plots that I obtained many years ago showing presence of primer dimers here with a lower temperature compared to the specific signal. So it's important. Let me maybe explain this to you. This is a typical real-time PCR plot in which we are looking at the intensity of fluorescence over time. The time here is expressed in terms of cycle. These are the PCR cycles, and typically we perform 40 cycles for a real-time PCR reaction. Nothing happens during about 15 cycles, and then at about between 15 and 20 cycles, we start to see the newly synthesized DNA by virtue of increase of fluorescence. And then it plateaus as the reagents are depleted or the reaction saturates. So primer dimers are not visible with this type of plot, but if you use CyberGreen, you have to check for the presence of primer dimers, and this is done by adding an additional step at the end of the PCR, in which after the 2 hours of PCR, you basically decrease the temperature, the experimental temperature slowly until the populations, the DNA populations separate, and that's called the Mel curve. And then monitor at which concentration the signal is changing. So that is the first method to detect DNA produced during the real-time PCR. The second approach is to use a TaqMan probe. So here, of course, the PCR reaction is still initiated with a 5' and a 3' primer that anneal to the complementary cDNA. But in addition, we add one more oligonucleotide to the mixture, which is a probe. A probe is a small oligonucleotide that is complementary to the target DNA, similar to the primers. And it's typically labeled to a dye, a fluorescent dye on one end, and to a quencher on the other end. So in normal conditions, when the probe is not is sitting around and there is no PCR amplification, the quencher quenches the fluorescent dye and there is no signal. During the PCR reaction, during the annealing of the PCR reaction, the primers will anneal and the probe will anneal, and the quencher will still quench the dye. However, as the Taq polymerase comes on and starts to generate the complementary strand of DNA, it will also well, the Taq polymerase is not only a polymerase, but it also has an exonuclease activity. And that exonuclease activity is going to chop off the probe and basically release the quencher, and the fluorescent dye will be able to emit fluorescence. It will be released and then start to emit fluorescence. So the advantage of this approach, there is increased specificity because the reaction specificity will be based not only on the primers but also on the probe, so it's like a double security if you want. It's possible to multiplex. You can have multiple probes that are labeled with multiple colors and multiple fluorochromes, so looking at different genes in the same reaction. And it's not detecting primer dimers. So now, after you have established an assay, whether it's one or the other method, and you run your samples, you basically generate you basically generate plots that look like these here, and the analysis consists in using a threshold value, which is here represented by a red line, and calculating the number of cycles needed to reach this threshold value for each of the samples and each of the genes. And you do that for every gene and a housekeeping gene. A housekeeping gene is a gene that is present in that cell that is not supposed to be affected by the treatment or the activation that you are applying to the cell. It could be a ribosomal protein, it could be beta-actin, this type of genes. And then you have to normalize, you have to basically report the expression of the gene of interest to the expression of the housekeeping gene. And you can do that in two ways. You can either generate absolute data, absolute values using a standard curve, or you can use a relative expression. The absolute quantification is done by generating a standard curve for each of the genes that are studied, and by serial dilutions of the standard, obtaining the absolute concentration in whatever units you're using of the target gene in your sample of interest, and then that allows you to have an absolute quantification. However, this is very difficult to do. First you need purified DNA for each of the genes, and accurate DNA quantification is of course key, and pipetting and reproducibility, all of these are difficult to achieve. I found that as a proxy, some people use cDNA from one unrelated marker gene, and then therefore generate only one standard curve and use that standard curve to quantify all the genes. So this is like an intermediary approach, if you want. The approach that most people use, and is really the easiest one, is to express the data relatively. And this is a method called the ddCt method, in which first we take the threshold value for the gene of interest and the threshold value for the housekeeping gene, calculate the delta between those two values, and then do the same thing for a control sample. For example, resting cells compared to activated cells. And so that's a second subtraction, a second delta that is calculated here. And then a mathematical transformation of this double delta will take it to a linear value. And that linear value corresponds to a fold increase of gene expression. So I will show you the data that we've generated using this equation here. And then I wanted to just mention this. This is a very new approach that I found published in 2011 of a single molecule direct RNA sequencing without cDNA synthesis. So everything that we've heard about so far was obtained after reverse transcribing mRNA in cDNA, and that can introduce biases and artifacts in the system. So this publication came up with a technique to sequence RNA directly. So we have to follow up on this approach in the future. So again, a comparison of all of these techniques. The cDNA microarray technique is high throughput. The genes can be known or not known. We'll talk about this later. It is not absolutely quantitative. It's semi-quantitative. And cDNA synthesis is required, as well as for the next two. RNA-Seq is high throughput and quantitative. qPCR is really quantitative, not high throughput. So before I move to the application part of the talk, I wanted to ask if you have any questions on the technique. No? So now let's see how those techniques can be used in the context of xenotransplantation, and I will show you 3 examples. How are we doing on time? That should be okay. So as you heard this morning and in prior sessions, xenotransplantation is transplantation of organs across species. is limited by the occurrence of a very strong hyperacute rejection that's triggered by the binding of antibodies, which in turn activate complement endothelial cells and lead to coagulation activation, and also to platelet and neutrophil sequestration. We've been using pigs that are genetically modified that lack the alpha-galactosyl transferase that's responsible for the expression of the alpha-gal epitope that's binding about 80% of the antibodies in the human blood, and CD46 or CD55 is used to inhibit complement that's activated by the remaining non-gal antibodies. You've already seen that. Just to set the stage on the samples that we've analyzed for gene expression, I wanted to show you basically the results we've obtained within the last 5 years with the pigs that were either gal knockout or gal knockout CD46 in our xenogenic lung perfusion model. So whereas a wild-type lung rejects within minutes of perfusion, a gal knockout lung shows a prolongation of function, which is slightly improved by expression of CD46. But not yet to the level of an autologous lung, which is a pig lung perfused with pig blood. So our lungs fail within a few hours, even with those genetic modifications. And we wanted to know what are the genes associated with this injury in our model. Thrombin formation is evident, as measured here by an increase in plasma F1+2 levels. Even in a gal knockout or a gal knockout CD46 lung perfusion, we still see huge levels of thrombin. Of course, it's less than in a wild type, but this is still huge compared to a clinical situation. In a clinical situation, I think everything above probably 1 would be considered pathologic. And platelets disappear, about 50% of the platelets disappear from the perfusate within minutes of perfusion, even with those genetic modifications. So we wanted to look at genes that are expressed by either endothelial cells or macrophages in the lung, or by human blood cells, and try to see what genes are upregulated during this perfusion. So the genes we looked at so far are genes that belong to the coagulation pathway. So we looked at tissue factor, which is the initiator of the extrinsic coagulation pathway. We looked at some genes involved in the fibrinolysis process. Selectins, adhesion molecules, complement and thrombin receptors involved in inflammation. And then we tested a panel of housekeeping genes, and we looked at a variation between normal and perfused lungs, and we identified RPL32, which is a ribosomal protein, as being the best housekeeping gene in that it showed very little difference between normal and perfused lungs. We also looked at a few human genes, both housekeeping and TLR4 and Agar, which is a receptor for HMGB1, to try to look what was going on the human scene. So all of this was done by real-time PCR. So we're here looking at expression of pig tissue factor. After 30 minutes, we see a very few changes in expression, as we could expect in this short interval for tissue factor. So on the blue shows expression levels in a wild-type lung, the red in a gal knockout, and the white in a gal knockout CD46 lung. And we're looking here at pig tissue factor. So we have primers and probe that can pick up pig tissue factor, but not human tissue factor. When we look at 4 hours after perfusion, we see an increase in pig tissue factor in all groups, including the autologous groups. So some of this injury is probably mediated by ischemia or perfusion and not only by xenogenic stimulation. So definitely we have an increase in expression in tissue factors, so we know that the extrinsic pathway of coagulation will be playing a role here. We also looked at fibrinolysis, genes involved in fibrinolysis. This is a schematic of the molecules involved. And here, again, we see an increase, and this is shown at 4 hours after perfusion in all groups for all those molecules. It seems, for example, if we look at tissue plasminogen activator, that we might have a decreased expression in a xenogenic situation versus an autologous. Of course, this has to be confirmed with a higher sample size. But this is suggesting that perhaps there is also a defect in the regulation of fibrinolysis in addition of coagulation, and very little is known about this in our field so far, so we probably should look into that as well. As far as inflammation, several of the genes that we looked at were not, their expression was not changed within 4 hours of perfusion, and some had inconsistent changes. E-Selectin, which is an adhesion molecule expressed by endothelial cells, was upregulated within 4 hours of perfusion. So definitely this is likely to play a role in cell trafficking and transmigration of cells within that time frame. Well, this is at the mRNA level, but we still have to look at the protein level, whether it's expressed in that time frame. And then looking at human genes, we found a very strong upregulation of human TLR4 within minutes of perfusion. We went back to 10 minutes, and we see very high levels even at 10 minutes. So we thought that this maybe is reflecting the cellular infiltration of the pig lung with human blood rather than upregulation of those genes. And I will show you I think the data with housekeeping gene. We have similar results with another human gene, agar. And then when we look at the housekeeping gene, the human housekeeping gene, we also see this increase. So basically, and when we calculate a ratio, we have a similar profile. So basically, we think that the earlier human genes that we looked at were reflecting more cellular infiltration than upregulation. So in summary, we see an increase in the expression of tissue factor, fibrinolysis involved genes, L-selectin, and human genes after perfusion of a GAL knockout CD46 lang. We also have some data looking at chemokines and cytokines, and we're completing this profile. So the second example I wanted to show you is a study that was done by Simon Robson a couple of years ago using a DNA array study, and I think with validation of the results with real-time PCR, I believe. The study was designed to compare the expression of a known set of genes in cardiac and kidney xenografts. This is the list of the, I think, 64 genes that were studied. So this is now a DNA microarray that was customized with pig probes for a defined set of genes that was selected based on their potential involvement in rejection. Those genes are mostly expressed in the vasculature, I think. Not entirely, but they are involved in vascular biology. And Simon can add if there is anything to say there. This is a figure of the paper showing what the real data look like. So here you can see a picture of a chip. So we here have 6 examples. The top 3 chips correspond to a heart xenograft. At the bottom, it's a kidney xenograft. The C corresponds to controls, and R non-rejected xenografts, and R rejected xenografts. And among the 64 genes that were studied, I think about 18 came out positive with different samples. The results were confirmed here by Northern analysis, in which RNA is probed with, is revealed using probes. And then also the results were confirmed by immunohistochemistry to basically confirm that the increase in mRNA was corresponding to an increase at the protein level. And so this is something to keep in mind whenever you look at mRNA levels. If you really want to know if it has an effect in the biology of your system, you also have to verify that there is a corresponding increase in protein levels. And that's what was shown in this paper. And then the third example I wanted to show you is a paper by Jerry Byrne, published in 2011, in which they looked at gene expression in cardiac xenografts. Those cardiac xenografts were all from orthotopic transplantation, which means that the pig of the heart of the baboon was replaced by a pig heart. This is a little bit different from what we do in our center, in which we transplant an additional pig heart in the abdomen of the baboon. But here the life of the baboon was dependent on the pig heart. And they looked at three different groups. Group A corresponded to a series of cardiac xenografts that were rejected. And you see here some coagulation related necrosis in a cardiac xenograft, which was lost, I think, around 3 weeks post-transplantation. Group B corresponded to cardiac xenografts that were explanted for other reasons, but they were not rejected. And you see here the typical histology of such a xenograft. And then group C corresponded to a perioperative graft failure after xenograft. This is a phenomenon that's seen especially in this model, in which some xenografts never function well, and they have to be explanted within days after transplantation. The platform that was used here was an Affymetrix platform. I believe pig Affymetrix platform, so basically interrogating the entire genome of the species and not assuming on specific genes or selecting specific genes. So the three groups are represented here in this Venn diagram. And as you can see, there are many genes that are upregulated in one or the other group compared to a normal sample, but very few genes are upregulated in all three groups, differentially upregulated. There are a few here in the middle. This table shows the major changes in gene expression that were detected in this study for either rejected, surviving, or early failing grafts, with either an increase in expression or a decrease in expression. Another way to look at the same data. And at the end, when the results were classified per pathway, per biological pathway, if you want, it was found that the changes were found in genes involved in injury pathways, inflammation pathways, as well as metabolism and other pathways. So many different pathways were actually affected by the xenograft injury. I don't know whether within this series there were genes involved in carbohydrate synthesis, GAG synthesis, but it would be interesting to look up whether any of those enzymes that you are using or are involved in synthesis of GAGs were interrogated during this study. This is just showing how systems biology integrates genomics as well as other omics studies. And if you have questions or want to follow up on any of these approaches, you can email me. I'd like to thank the team at University of Maryland, our colleagues all over the world, in Pittsburgh, in Boston, Harry Dawson, who helped with our real-time PCR assays. The work is supported by NIH United Therapeutics, and I will stop here. Yes?
Whenever you looked at your genes of interest for your gal knockout,
Mm-hmm.
you used the qPCR technique, correct?
Yes. Yep.
Have you thought about going back and using a non-targeted approach
Yes.
to see what you might find that you don't expect?
Yes. Yes, we are trying to do this. We performed an experiment with the aim to do this specifically a few weeks ago. We happen to have at our university an institute of genome sciences with people who do RNA-Seq on a daily basis. So with a collaboration with United Therapeutics, this is something that we are hoping to be able to achieve. And then also, so this will interrogate the entire mRNA species that are available. But also in between, we've also selected our best RNA samples and sent them to Simon Robson to be analyzed on the array study that they used. So we are hoping to learn also from these studies.
And Yeshiva is looking at the whole transcriptome as well.
Hmm?
Yeshiva will be looking at the whole transcriptome of the result.
Yushi.
Honey Yeshiva. The co-author who made those mini arrays, which were just targeting the differences between the vascular response of
Yes.
the kidney and the heart
So she will be looking at the whole pig transcriptome.
of those samples.
Okay. So, any other questions? To me, I didn't make any slides about this, but to me, what really would be interesting within this grant, within this project that we have, collaboration with this group, would be to look at the specific expression of enzymes that are involved in carbohydrate synthesis. Transferases, sialidases, all the enzymes that you use for the xyloside, for example, we could, by real-time PCR or another approach, we could look at their expression during xenograft injury or rejection and maybe try to correct if there is an imbalance after rejection.
You already have result with selectin, right?
Yes.
Selectin, I thought.
Yeah, we do have
There has to be something.
selectin is increased, yeah. All the NF-kappa B dependent genes are upregulated. We know we have thrombin. Thrombin, that's something also that Simon Robson showed, is activating NF-kappa B dependent genes in endothelial cells. This was shown maybe 10 years ago. And so we expect to see upregulation of those genes because we have thrombin formation.